Much, and thank you for inviting me as well. I will just share my presentation, which has disappeared. Typical. Give me 1 second. There we go, found it. Sorry about that. Can you see that?
Yeah.
Brilliant. So yes, again, thank you very much. I have been a postdoc now for about just over a year, but what I'm going to talk about today is the stuff that I did in the first half of my PhD, which was at the University of Oxford, because I thought that it might tie in a little bit better with what you've been studying in the course. So I'll start off by probably explaining something you hopefully already know. The ocean biological carbon pump involves the transport of organic matter from the surface ocean to the deeper ocean. And it does this by photosynthesizing phytoplankton near the surface, take up carbon into their bodies, get grazed on by zooplankton, and then when they die, they sink through the water column. And as they sink, they get remineralized back into their dissolved components. But some of these particles or detritus won't get remineralized until they reach deeper down in the ocean, meaning the carbon in the particles can get locked away in the deep ocean for up to 1,000 years. So we have this vertical transport of carbon from the surface ocean to the deep. And this has been shown to really regulate the atmospheric carbon dioxide, because there was a cool study in 2006 where they had an ocean biogeochemical model where they turned off all of the iron, so effectively shutting off the biological carbon pump, and the atmospheric carbon dioxide went up by about 200 parts per million. So the biological carbon pump is very important for our climate, and therefore very important to model it correctly in the biogeochemical components of Earth system models that we use to predict future climate change and inform policymakers in IPCC reports. So a lot of my PhD was trying to have a look at the parameters that we use in the models associated with the biological carbon pump and how to improve them so that we get model outputs closer to observations and therefore a better model. So ocean biogeochemical models essentially try and take all of this complex stuff on the right and simplify it into something feasible that we can model. So a lot of them build on this NPZD structure, where we have 4 boxes, one for the nutrient that will be the core of the model, for example, something like nitrate or phosphate. And then you have a phytoplankton box, zooplankton, and detritus. And stuff can get shared or transported between these boxes, depending on whether there's grazing or mortality or remineralization. And this forms the base of a lot of biogeochemical models. And the more complex the model, maybe the more boxes and interactions there are. But in this simplified version of the complex stuff that's all shown on the right, we've had to simplify a lot of processes and systems. And therefore we have these parameter values in the models, which are pretending or representing some process on the right. And these are the parameter values that we could tune or optimize in the models to get a better version of the model. And one example of these parameters is the Martin B parameter. And this B parameter down here in this equation controls the amount of remineralization occurring on sinking particles in the model. So if you have a let's start off with a low B here at 1.5. We follow this blue line, so we've got lots of sinking flux at the surface, and then a lot of it gets remineralized in the upper water column so that there's less flux sinking to depth. So this would mean that we have a weaker biological carbon pump because we have less downward transport of organic matter. But then if we had a lower B, 0.4, we follow this red line. Again, we've got high flux at the surface, but a lot more of this flux is reaching deeper into the ocean. So we have a stronger biological carbon pump. And the default value in a lot of biogeochemical models, which uses this Martin B parameter, is 0.858, based on only 6 profiles in the Pacific Ocean taken by Martin and others in 1987. So it's a small sample of the ocean, but we apply this single value to the global ocean in biogeochemical models. And there was a study in 2009 where they varied this B parameter to see its effect on atmospheric carbon dioxide. And varying it from 0.4 to 1.4 can have a massive effect on the atmospheric carbon dioxide. So we need to make sure that this value is correct in ocean biogeochemical models, because it can have a massive influence on our global climate. So it's a very influential parameter, but it's 0.858 in the models, but it's been measured to be anything from -3 to above 3 in the ocean with observations of flux. So on the left, we've got B values observed in the ocean according to flux observations. And I've plotted them with latitude here. And you can see that there's a big spread in what B has been measured to be. But on the right here, we've got temperature-dependent parameterizations, or modeled B, by 2 different contrasting past studies, one by Henson and others in 2012, which is this blue line, and one by Marseille and others in 2015, which is this red line. And they're both completely polar opposites, showing a spatial B pattern, which is dependent on temperature. And the observations don't even really fit with either of them. They're in the middle and they've got a completely different structure. So we obviously don't really understand this B parameter. And so can we even use 0.858 in our models? So I've explained that there are very influential parameters in biogeochemical models, and there's a lot of scope for us to improve them by parameter optimization. So in the first 2 years of my PhD, I focused on the MOPS biogeochemical model. And this, again, if you recognize this NP-DZ structure, it's just got a few more boxes because it includes the oxygen cycle and the denitrification and nitrogen fixation. And so I use this and tune some parameters in this model. And if you'd like to know a little bit more about MOPS, Iris Kreis actually gave a talk at this symposium 2 years ago, and her video is on YouTube, so I definitely recommend if you're interested in MOPS. So before we can improve the model, we first need to know how well it does. So we need to define model performance. And this can be done by calculating a misfit, or F, which is some root mean square error between the model outputs and real observations. And these observations can be oxygen, nitrate, phosphate, which are quite well sampled in the ocean now and available in databases such as World Ocean Atlas. So say we wanted to tweak or tune 3 parameters in the model, maybe one of them the Martin B parameter. It means we have a misfit function, which is 3-dimensional in this three-dimensional parameter space. And within this space, we have a minimum in the function where it coincides with a parameter configuration that gives you the best version of your model. So you'll have peaks and troughs in this misfit function. And the peaks are where you have a really bad version of your model. You don't want to use those parameters. And then minimums where you have a good configuration of your model. And it's the best minimum that we want to find, the lowest of lows. And MOPS has lots of parameters, not as many as more complex models, but we chose 6 to optimize. One of them was the Martin B parameter. And the process of optimization is this loop here. So you have some initial parameter configuration, maybe with the default values of 0.858 for the Martin B. You then run your model, in this case MOPS. You spin it up for 3,000 years so that it can equilibrate. And then you calculate your misfit to observations. That's this misfit for performance. And this misfit is then given into an optimization algorithm. And it will, in an intentional and clever way, work out a new set of parameters for you to try, which will hopefully guarantee a better misfit than the one before. So it's not just some guess, it's actually intentionally trying to reduce your misfit every time you loop. So then you input these new parameters into the model and you keep looping until you converge onto a small misfit. And there are lots of optimization algorithms out there, but the one that had been used before on MOPS was by Iris Christ in 2017. And they used the CMAS, which is one algorithm which I won't explain now, but it used about 1,710 MOPS evaluations to successfully optimize 6 parameters. So I'll explain the top left plot first. On the x-axis, you have evaluations. So one evaluation is one run of the MOPS model. And then here we have the misfit value. And you can see through the number of evaluations, every iteration of the model, of the model optimization process, we get a reduction in the misfit. So we're getting closer to observations. And this is because we're converging on the optimal parameter values. So down here, we've got 6 subplots. Each of them show the parameter value for each of the 6 parameters. And the hashed lines show the optimal parameter value for each of these 6 parameters. And we're actually optimizing to synthetic observations in this case. So we know where our optimal values are, just to make sure that we know the optimizer is doing well. And you can see with time or with evaluations, we converge onto these 6 optimal values within this number of MOPS evaluations. And that's quite a lot. It was successful and it was great, but it's quite a lot of evaluations, especially if you have a model that takes a long time to run. So the point of the first half of my PhD was try and do the same here, but do it quicker and more efficiently. And so I repeated what Iris did in 2017, but with a new optimizer that came out of the Maths Institute at the University of Oxford called DFOLS. And we managed to do not quite as well as what they did, but we managed to do it in 42 evaluations of MOPS. So again, the same plots as on the left, but we've got the reduction in misfit over a lot fewer evaluations, and we converge on 6 parameters, but not always this one. So this one didn't do as well, unfortunately, but because the model isn't very sensitive to this parameter. So we sort of pushed that under the rug and said that that's not too bad. And the fact that we've managed to get a computational saving of 2 orders of magnitude, means that that's also not too bad. You've got wins and losses every time you play with stuff like this. So this was a massive computational saving. And if you're interested in this, we did publish in D&D. But after I'd done this, I then thought, can we make it even quicker? I know it's crazy quick already, but there's still some space to try and be a little bit more efficient with this. And the more efficient we are with parameter optimization, the more encouraged developers are to systematically calibrate their models to observations. So the next thing I tried was to try and optimize with a shorter spin-up. So I mentioned before that we had to run MOPS for 3,000 years before we could calculate the misfit. And that's so that the MOPS model can equilibrate throughout the global ocean so that traces stop changing with time. So you're in a steady state. And it needs to be over 3,000 years to do this because of the long timescales of the deeper ocean. So if you have sinking organic matter in the North Atlantic and it reaches the deep Atlantic, you then need to give the model enough time for this matter to travel all the way along the base of the global ocean conveyor belt, where it passes through the Atlantic, the Indian Ocean, and ends up in the deep North Pacific. So you need to see all of that happen to be able to get to an equilibrated state. And again, Iris, in her YouTube talks about spin-up. So if you're interested in that, I'd still recommend you have a look at that video. So my question here was, do we need to be equilibrated to still optimize successfully? So I tried, and we sort of failed. But there was a pro that came out of this. It wasn't all cons. So the left here is again similar to the plots I've shown before, where we have evaluations of MOPS on the x-axis and the misfit on the y-axis. And I did 4 experiments optimizing again those 6 parameters. And the pink line here with the diamonds was the MOPS model with a 3,000-year spin-up. The red one is with a 2,000-year spin-up, and then we have 1,500-year spin-up. And we had a successful optimization with the 3,000-year spin-up as before, and the 2,000-year spin-up also was successful. We managed to converge to the optimal parameter values, and we got the misfit below observational noise. But the problem comes when you go shorter than 2,000 years. These did not manage to optimize at all. DFLS, the optimizer, didn't know what to do. And I tried to understand why, and it's to do with this equilibration of the deep timescales, the long timescales of the deep ocean. So up here on the right, these 4 plots here, I'm just going to explain this one first. I evaluated the MOPS model at multiple locations within the 6-dimensional parameter space and calculated the misfit. So here we have the misfit values after 3,000 years and the misfit values after 2,000 years. And you can see this nice, easy, linear relationship. And that's what we want to see. If we have a linear relationship between these 2 axes, it means that a shorter spin-up, or this shorter spin-up, will give us successful optimization. The problem comes when we get into shorter spin-ups. So we've still got 3,000-year misfit here on the x-axis, but we've got a 1,000-year spin-up on the y-axis. And we've now got this non-linear tail coming into the relationship, which gets more pronounced with a shorter and shorter spin-up. And this non-linear tail is what threw off DFLS and made it struggle to converge onto the correct optimal values. And as I alluded to this parameter before, this nonlinear tail coincides with low values of the Martin B parameter. And so if you remember before, low values of Martin B means that we have essentially fast sinking particles. So we have more transfer of detritus to the deeper ocean, where we have longer timescales. And all of that organic matter needs to travel all the way to the deep North Pacific. If we had a high V, like up here, we got slow sinking, we've got lots of remineralization happening in the surface, and all of that sinking organic matter stays in the surface. And where we have quick overturning timescales, so we don't actually need too long a spin up to be able to have successful optimization. But it's when we have fast sinking particles when we have this issue. So we failed to successfully optimize shorter than 2000 year spin up, but we still got a computational saving of a third, if you think about it. So not too bad. And it's not all doom and gloom, because it might depend on it does depend on your initial conditions of the model. So on the left is what I showed before, which was this bottom left here. And on the right is now where we initiate the model with the same tracer distributions as the observations that we're using for calibration. So we start off with a very good estimation of what we're trying to get to. And we can successfully optimize on very, very short spin-ups in this time. But the caveat here is we're still calibrating to synthetic observations. So if we were to translate this into the real world, it might not translate very well, because we're never going to get an initial state of the ocean for all of the traces the model needs that is exactly the same as our target observations. So I haven't quite got through this question yet, but it's food for thought and is hopeful as well. So in summary, ocean biogeochemical optimization is computationally expensive. There's a lot of parameters in biogeochemical models that are very influential to the biological carbon pump, but we don't actually know what specific value they should be set to. But if we can systematically optimize more easily, then we can use observations to actually constrain those parameters for us. We tested a very efficient optimizer and reduced the computational expense of optimization by 2 orders of magnitude, and then reduced it by another third for MOPS by showing that we don't need to spin up fully to a steady state, but can get away with 2000 years, and may even in the future be able to go shorter if we have a better guess of the initial state of the ocean. But thank you very much, and I'm open to any questions.